I'd like to continue our discussion of the diversity of life, and in particular pick up on our discussion of plants. And today we'll continue with the subkingdom Angiospermae. So last time we talked about the gymnosperms, and now I'd like to talk about the angiosperms. And the angiosperms are flowering plants. The only division within the Angiospermae is the division Anthophyta, and so these are the flowering plants. So these are the most advanced of the plants, most recently evolved, and the most successful group of plants. And most of the plants that we eat are flowering plants, or the products of flowering plants. So we're going to see some similarities to the life cycles we talked about already, but in looking at general characteristics, again, the division Anthophyta would be the flowering plants. Seeds will develop within a fruit. So that's one of the definitions of the flowering plants, is not only that they have a flower, but that seeds develop within a fruit. And the fruit is derived from the flower. It's going to be derived from the ovary or carpel or pistil. These are interchangeable terms of the flower. Another way of defining the flowering plants is that they practice what's known as double fertilization, and we'll get to that in a minute, but this means that both of the sperm that are produced by the pollen grain will actually have a function here. And they produce a triploid endosperm instead of the haploid endosperm that we saw in the gymnosperms. So again, if I would suggest you look at the overall life cycle, review the life cycle of gymnosperms before we get into the life cycle of the flowering plants. So the key anatomical feature of flowering plants, of course, is the flower. And as we look at the flower, there are sex organs, and then there are what we call accessory structures as well. So looking at the reproductive structures then, if we look at the female reproductive organ, it's called the carpel, also called the pistil, P-I-S-T-I-L. And there are three parts to the carpel or pistil, one being the stigma. The stigma would be the top of the carpel or the pistil. The style, there's usually kind of a neck-like region, and that's known as the style. And then there's typically an enlarged region at the base of the carpel, and that's the ovary. So it's the stigma, the style, and the ovary collectively make up the carpel or pistil. Now, a lot of times the term carpel is just used interchangeably with ovary, and so sometimes that can be a little confusing. So just be aware of that. And then it's within the ovary then that we will have ovules, the oval mass of tissues that ultimately give rise to the seed. So again, we're going to be looking at the same concept here, that one ovule generates one seed. So that's the female reproductive organ. The male reproductive organ is called the stamen, and there are two parts to the stamen: the filament, which is basically a support structure, and the anther. And it's within the anther then that the pollen grains are produced. So that would be the male reproductive organ. And then in addition to the reproductive organs, a flower typically has what we call accessory structures, and some of those would be petals. And so the petals are most typically a pigmented portion of the flower. And collectively, we refer to the petals as the corolla. And then typically, again, there will be more leafy-like structures associated with the flower. As the flower is growing, they would help encapsulate the bud, and these are called sepals. And collectively, the sepals are referred to as a calyx. So in other words, you could have a corolla of 6 petals, you could have a calyx of 6 sepals. Okay, so again, both the petals and the sepals, and also the base of the flower is typically referred to as the receptacle. All of those would be considered accessory structures, meaning they're not absolutely required for reproduction, whereas the sexual organs are. Now, it is the ovary of the flower then that actually develops into fruit. So when we look at fruits, fruits contain seeds. The seeds were originally ovules within an ovary, and then the ovary develops into the fruit. And so again, fruits sometimes are succulent or fleshy, but there can also be dry fruits, as we have in nuts and so on. So again, there's a lot of variety that we actually see in both seeds and fruits. But again, seeds develop from ovules, which were within the ovary, and so seeds then develop within a fruit. So again, be sure that you review the overall life cycle, because we're basically going to be looking at this overall life cycle of gametophyte and sporophyte generations. And so again, same general concept, but we're looking now specifically at flowering plants. So let's go ahead and then see how flowering plants do reproduce. So looking first at kind of the male reproductive organ, so we'll look at the anther. It's within the anther that actually the pollen grains develop. And so again, we're looking at similar terms that we used before and talking about the gymnosperms. So we will have essentially immature pollen grains develop, so-called microspores that will develop within the anther. And then these will divide once, to produce a tube and generative nucleus. And at that point then, this is the mature pollen grain or microgametophyte. So microspores develop into microgametophytes. And then the microgametophytes then must be taken to the female part of the plant, taken to the carpel or the pistil. And that can be by wind or actually by a pollinator, and I'll get back to that in a second. So here's a slide showing us a cross-section through an anther, and we can see pollen grains within. And again, if the pollen grains only have a single nucleus, then those would be microspores. And again, once they've completed one division, if we're seeing two nuclei or two cells, then those would be microgametophytes. And again, one would be a tube nucleus, one is a generative nucleus, which will eventually give rise to sperm. Now, meanwhile, in the female portion of the plant, within the carpel or the pistil then, an ovule will develop. A single megaspore will be produced within that, and the megaspore will divide into a megagametophyte. And if ever there was really an oxymoron, this is that example, because mega means big, so megagametophyte, it would imply it's a big gametophyte. But here the megagametophyte is only 8 cells in size. So again, as we've gone from primitive plants to more advanced plants, we've seen the gametophyte get smaller and smaller and smaller here. And so again, it's really only 8 cells. And so we're going to be interested in 3 of these cells, the ovum and the so-called polar nuclei or polar cells. And those will both be involved in this reproductive process, but we'll come back to that in a second. Okay, so pollen grains then need to get to the stigma of a flower. And so again, in the flowering plants, sometimes wind does that, especially with things like grasses. But more typically, what's happened is that the flowering plants have co-evolved with insects and other animals, and animals then act as pollinators. They'll transport the pollen, and actually, in looking for nectar and other sweets on a flower, they'll actually end up depositing pollen grains on the stigma. Once there, pollen grains then can grow, and so the pollen grain will form a pollen tube, as we've talked about before, and there will be a tube nucleus guiding the growth of the tube. And then the other nucleus, the generative nucleus, will divide to form 2 sperms. So this diagram is showing us the sperm nuclei. It's neglecting to show us the tube nucleus, unfortunately. And of course, the ovary could have a number of ovules. This diagram is just showing us a single ovule. And within that ovule, we'll have our megagametophyte with the 2 polar nuclei and with the ovum. And again, in a cross-section of the ovary, again, these are what the ovules look like. And so you can actually see the megagametophyte within the ovules. And so again, the pollen grain has to form a pollen tube to extend down and get to the ovule. So eventually that pollen tube grows down. The 2 sperm nuclei then have access to the megagametophyte. One of those will fertilize the ovum to form a zygote. And that zygote will then divide to form an embryonic plant. And this is where the so-called double fertilization comes in. The other sperm nucleus will fuse with the 2 polar nuclei. And so these are all haploid cells, remember. So the gametophyte is haploid. So if you have 1 N plus 2 N and another N, that's 3 N. So this will form a triploid cell, a 3 N cell. That will divide and eventually form the endosperm of the seed. So in flowering plants then, they have endosperm, which provides nutrition to the developing embryo, but it's a triploid endosperm. So this is referred to as double fertilization because both sperm are used. I don't really love the term because really the only real fertilization is fertilization of the egg and sperm. That fusion of those 2 gametes, that's what fertilization is. That produces the zygote. That produces the embryo, the offspring for the next generation. So anyway, but that's the term they use. So again, we'll end up with a triploid cell and triploid endosperm, and then our zygote, which will develop into the embryo. And then the surrounding diploid tissue of the ovule will eventually form the seed coat of the seed. So again, same idea, 1 ovule translates to 1 seed. So again, the role of the nuclei again. So we have a tube nucleus and we have a generative nucleus. And the pollen tube will extend. Tube will basically control the elongation of that tube. The generative nucleus will divide to form 2 sperm nuclei. And then when we look at the roles of these sperm nuclei and the tube nucleus will degenerate. It will not play a role in the life cycle. One of the sperm nuclei will fertilize the ovum. That will form a diploid cell. That diploid cell will then is the zygote. It will mitotically divide and differentiate and eventually form the embryonic plant within the seed. The other sperm nucleus will fuse with the 2 polar nuclei. So if we have 3 haploid nuclei and they fuse together, that forms a triploid cell. And that triploid cell then will mitotically divide to form the endosperm of the seed. And recall that endosperm stores nutrients and provides those nutrients to the growing embryo when the seed germinates. So again, this kind of encapsulates all the things we've talked about then. I don't think I'll go through it all again, but one of the things I want to point out is that an ovary can have a number of ovules. So each ovule then that's fertilized requires a different pollen grain. So one pollen grain will grow to a single ovule, and that will generate a single seed. So when you open up a fruit, if you see dozens or even sometimes hundreds of seeds inside, that means that in the original flower, that took hundreds of pollen grains to actually fertilize each one of those ovules and produce each one of those seeds. And again, when we look at the final seed product then, we end up with the diploid embryonic plant, we end up with a triploid endosperm, and the surrounding ovule diploid tissue will develop into the seed coats. So this is a cross-section through a seed. And they would be developing then within the ovary, and so the ovary then forms the fruit. Sometimes it's a succulent or fleshy fruit, sometimes it's a dry fruit. So again, we've already talked about fruits then contain seeds. So that's kind of the definition of a fruit. And if you're eating something that's a vegetable, it's a quote-unquote vegetative part of the plant, which would be the non-reproductive part of the plant. Okay, now, within the Anthophyta, there are 2 classes. One would be the monocots or monocotyledoni, and the other the dicots, sometimes called, or the eudicots, and it would be the class dicotyledoni. And they take their names from the fact that the embryo, the embryonic plant, has a structure known as a cotyledon. And the cotyledon is the part of the embryo that absorbs nutrients from the endosperm. So it becomes the interface between the embryo and the endosperm. It absorbs the nutrients and then distributes those nutrients to the rest of the embryo. So what defines the monocot from a dicot ultimately comes down to the seed. Monocots have a single cotyledon, whereas dicots, or the eudicots, will have 2 cotyledons. So you can see in this particular plant that I showed you before, we see 2 cotyledons, and so this would be a dicot plant. Beans, peas also would be examples of dicots. So basically, if you're eating a seed and it will divide into 2 halves for you, you're eating a dicot. Now, we can't always look at the seed, so there are some indirect ways to try to determine whether you're looking at a plant that's a monocot or a dicot. So again, the seed would be one place to look. Also, you can look at the leaves. Monocots tend to have parallel veins in the leaves, whereas dicots tend to have a netted type of venation. If you look at the stem and cross-section, the monocots will have the vascular tissue scattered in the stem, whereas if it's an herbaceous stem, the dicots will have the vascular tissues arranged around the periphery of the stem. Also, the root system in monocots tends to be fibrous, whereas in dicots there's generally a central taproot in addition to fibrous roots. The pollen grains will be different. Monocot pollen has a single opening, whereas the dicots generally have 3 openings. And then also when you look at flower parts, monocots tend to have flower parts such as petals or stamens and even carpels in multiples of 3. Dicots tend to have petals, sepals, stamens, and pistils or carpels in multiples of either 4 or 5. So again, these are kind of indirect indices of whether it's monocot or a dicot. Ultimately, it's the seed that defines whether it's a monocot or a dicot. And again, as I mentioned, fruits will be the reproductive part of a plant. Quote-unquote vegetables are the vegetative parts of the plant. So the part of a plant that's above ground we describe as the shoot or shoot system. And then, of course, the root system. And the stem, you may recall, has vascular tissues either around the periphery or scattered. The root will have vascular tissues in the center. And then as we look at the root system, places where leaves branch off or side branches come off of the central stem, we refer to that as a node. The regions in between the nodes are internodes. And of course, the leaf itself will consist of a blade as well as a petiole. So again, one of the aspects of this unit is that everything that you eat, at least that's not meat, you can kind of look at it and try to figure out what part of the plant am I eating. Is it sporophyte? Is it gametophyte? Is it haploid? Is it diploid? Where am I in this life cycle? And so you can kind of quiz yourself over and over and over again while you're eating. Okay, and again, sometimes parts of plants are a little confusing to figure out what you're eating. So, for example, an onion actually is a modified bud, so those are actually storage leaves. So an onion, these are actually leaves that you're eating. Something like a potato, that's actually an underground stem, believe it or not. So it's not always easy to tell what part of a plant you're eating, unfortunately. But again, if there are no seeds, at least you know it's a vegetative part. And even something like roots, sometimes roots aren't always underground. So sometimes, just as we can have underground stems, we can have above ground roots as well. So anyway, you really have to watch a plant a lot of times grow from seed to figure out exactly what part of a plant you eventually eat, or what part of a plant develops from a particular part of a flower. And of course, things like beets and so on, we would be eating a root there. Okay, now I want to transition now, getting back to our four super kingdoms, the Uniconta. We've already talked about the fungi, and now I want to begin to talk about the animals, which is the kingdom Metazoa or Animalia. So again, the kingdom Metazoa consists of organisms that are multicellular, heterotrophic, and the cells lack a cell wall. And of course, we are an example of a metazoan. And the metazoa evolved first in what's known as the Ediacaran time period. And sometimes this is referred to as the Ediacaran explosion. And this Ediacaran fauna was discovered first in Australia, the Ediacaran Hills of Australia, and that's where the name comes from. So we think that animals evolved in this time period, somewhere around 635 million years ago to 542 million years ago. Again, there's this explosion of animal diversity we see in the fossil record. And Ediacaran fossils have some fossil types that are unique to that time period that we don't see in any other kind of time period. And then the Ediacaran blends into what's known as the Cambrian time period, and that was originally known as the Cambrian explosion, and that's where we used to think that animal life first evolved, but we now know that it really evolved in the Ediacaran explosion. And so the Cambrian explosion is interesting because we still see a lot of representatives first showing up in the Cambrian time period that still exist today. Now, originally it was thought that the Ediacaran and Cambrian faunas were very different, but in some of the more recent discoveries, we now know that many of today's present animals actually first appear in the fossil record during the Ediacaran explosion. So you sometimes hear a lot about the Cambrian explosion, that the sudden appearance of animals occurring. It didn't really occur so much here as it did about 100 million years earlier in this Ediacaran time period. So we see animals evolving about 600 million years ago or so, maybe as much as 650 million years ago. And again, the Cambrian collection of animals is very distinct and different from the Ediacaran, although we are now finding some of these same Cambrian fossils in the Ediacaran time period. Now, in classifying animals, there are certain things that we look at, certain physical characteristics we look at, but actually the embryonic development of animals actually plays a role in how we classify animals. And so there are certain common features to animal development. Certainly we all start out as a zygote, diploid cell. And then the zygote begins to divide. In this early division, mitotic division, we refer to as cleavage. And later on, as the cells get smaller, we just refer to it as mitosis. But the zygote, as it divides, will form a solid mass of cells or a disk of cells called a morula. At some point, the cells rearrange themselves as they get smaller into a hollow sphere or hollow disk known as a blastula. And again, in most animals, that in turn transitions into an embryonic stage known as the gastrula, where a few things happen. The name comes from the fact that this is where the primitive gut begins to form. And then again, another series of stages will occur from that point on. But most animals exhibit these early stages, and then there are a lot of different pathways which can go on past the gastrula stage. Now, as we look at this then, so again, the zygote begins to divide through cleavage. Again, it will form a solid mass of cells, which is the morula. The hollow sphere or disk would be blastula. And then what happens is that cells begin to invaginate on one side and form a pocket. And this is the beginning of the gastrula stage. And so the gastrula stage, this is the beginning of the formation of the primitive gut, the primitive intestine. And in some organisms, it will stay just with one opening. And in other organisms, it will extend to the other side. Another opening will be produced, and then the intestine becomes a tube. So again, this is kind of visually what they look like, and I hope to be able to show you a little video of this as well in a little while. So the gastral stage is kind of important because a few things happen here. One is, I mentioned, this is the beginning of the gut formation, the intestine formation. And in some organisms, they'll have what we call a closed gut or closed digestive system. And examples would be something like, say, a sea anemone or jellyfish. So what that means basically is that this opening in the gastral stage, the blastopore, will develop into the mouth, and it will also function basically like an anus. So there's just a single opening into the gut. So if the anemone stuffs something in the gut, it will just digest what it can. Whatever it can't digest, it has to spit out the same opening. And the same kind of idea even in a jellyfish. So this would be a closed gut where there's just a single opening. And it's not the most efficient kind of a system, as we'll figure out later. An open gut is one that has two openings, both the mouth and an anus. And so again, what will happen in the gastral stage is that in some organisms, like you and I, two openings will be produced. And that original opening, known as the blastopore, has the potential to develop into the mouth, or it has the potential to develop into the anus. And so, again, this group would be known as the protostomates, this group are the deuterostomates, and you and I happen to be deuterostomates. And again, the open gut then means that there's both a mouth and an anus. So again, what it amounts to then is that you actually have a tube that runs through the body. And the good thing about that is that you can modify the tube along the way. So if you look at our digestive system, then we can have a place for storage of food. We can have a certain area where we actually digest the food. We can have another area where we actually absorb the food. And so that leads to much more efficiency, a much greater efficiency in digestion and absorption of nutrients. Now, another thing that happens in the gastrulation is that this is where the early tissue formation begins. And tissues would be groups of cells that have specific functions. So animals will produce what's known as ectoderm, which means the outer skin, an endoderm, and that will line the gut, the intestine, is the endoderm. And organisms that have only those two embryonic tissues are known as diploblastic animals. And then in more complex animals like you and I, a third layer of tissue called the mesoderm, the middle skin, if you will, also develops. And mesoderm eventually develops into things like muscle and bone and other portions of organs. And animals that produce all three of these embryonic tissues, ectoderm, endoderm, and mesoderm, are known as triploblastic animals. So again, the ectoderm typically develops into skin or outer coverings. Endoderm would be the lining of the digestive tract. Mesoderm would be muscle, bone, and those kinds of structures. Now, again, kind of looking at development of this mesodermal tissue, it can develop in some different ways. And then that also can eventually lead to body cavity development, which we'll come back to in a second. So again, another aspect of this gastrulation is that a closed cavity, body cavity, may form. And again, there's some variations here. So some organisms we describe as being acoelomic, and these are animals that do not have any kind of a body cavity within them. So if we're looking at something called a planarian flatworm, and we'll be discussing these in a future module, if we look here, the blue kind of represents ectodermal tissue, which would be the outer integument or skin. The yellow represents the endoderm lining the intestinal tract. So again, the intestinal tract is basically a tube here. And you'll notice that the mesodermal tissue is in red. But you'll notice that it's solid, so there's no space here, no cavity. So that would be an acoelomic animal. Coelom is a reference to a body cavity. A means without, so they are without a body cavity. Some animals have, we describe being pseudocoelomic, meaning they actually do have a space between the intestine and the outer body wall, but only the outer portion, only the body wall is lined by mesodermal tissue. And so mesodermal tissue, again, can be connective tissue. It can also be muscle tissue. So only the outer body wall is lined by muscle or connective tissue. The intestine is not. So they do have a body cavity, but it's not completely lined by mesoderm. And then the eucoelomic animals, like you and I, the truly coelomic animals, do have body cavities that are completely lined by mesodermal tissue. So the outer body wall and the intestinal tract is lined with mesodermal tissue. And again, that's a so-called true coelom. And the importance of the coelom basically is then that allows for organ development. It allows space for organs to develop. Because typically when cells are growing in animals, cells tend to grow until they contact other cells. It's something called contact inhibition. So if you have a solid mass of tissue, it would be hard for organ systems to evolve. But if you have a space there, then it allows for mutations that lead to development of new systems that are not inhibited by this contact inhibition. So it's thought to be an important evolutionary adaptation. Now, still one more factor that's important in classifying organisms would be symmetry. And symmetry would essentially establishes mirror images, running a plane through an organism and looking at mirror images. So some organisms we describe as being radially symmetrical, others bilaterally symmetrical. And looking at this then, a radially symmetrical animal is one that has multiple planes of symmetry. So something again like a sea anemone. We could divide it this way and have mirror images. We could divide it this way, have mirror images. We could divide it in a lot of different ways and have mirror images on either side. Bilaterally symmetrical animals like you and I have a single plane of symmetry. There's only one way that we can divide that animal to get mirror images of one another. And still other animals, sponges in particular, lack symmetry. So this is rare among the animal kingdom, but there are a few that lack symmetry. Now, I want to go back just real quickly, and again, because these early developmental stages will set the stage for whether we have some of these characteristics like symmetry and so on. So I'd like to show you a quick animation leading you from, and this would be of a sea urchin egg, going from fertilization through gastrula formation. And I'll just make a few comments about it, and I think it'll help you visualize this development. So what's happening here is that the sea urchin eggs have just been fertilized, and they're producing what's called a fertilization membrane to prevent multiple fertilizations. And you can see here, this would be the early cleavage that's going on. So the zygote has divided into two cells, and it will continue to divide. And this solid mass of cells then is called a morula. And the importance of these divisions is that it decreases cell size. Here we see a holosphere, so this is the blastula stage. And here we're beginning to see the beginning of gastrulation. Now, what's also happening here is that, again, we're also seeing the production of what will eventually be mesodermal tissue as well. So what we're going to see here is an indentation on one side, and this is the beginning of the primitive gut formation. So this opening is known as the blastopore. And again, in some animals that will be the only opening into the gut. In other animals, another opening will be produced up here, and that initial opening will either be the mouth or the anus. And then you can see these other cells coming off that are beginning to form the mesodermal tissue. So this will be endodermal tissue, ectodermal tissue out here, and mesodermal tissue here. So that lays a little bit of the foundation then for characteristics that we'll need to understand in classifying animals later. So see you next time.